Thanks, Marcel, and the organizers as well. So yeah, my name is Alvaro Martinez Barrio. I'm the Chief Data Analysis Officer at Pixelgen Technologies, and we are actually founded in Sweden with all meaning that does us for NF Core and also innovation in genomics. So I'm here to present you our innovation. It's called molecular pixelation, or shortened MPX, so I will refer to that a lot. And I hope that you find it as exciting as I find it. Right, let's dig in. So this is a cell, and in the left picture, you can see a cell in a normal confocal microscope. On the right, you can see the reconstruction that we have done of the MPX cell information to an idealized spheric projection. The cell, both cells, they are different. They have been treated with the same biological drug. In this picture, instead of showing the idealized form of the prior picture, we're actually showing the information of the MPX cell graph. So our native information are graphs. And what is important to see here is actually the cluster of receptors that you see there in red. So every node of the cell graph is colored by a certain receptor that this biological drug is targeting, actually. And we are able, in a couple of weeks, to reconstruct the mechanism of action of this drug and how the different receptors engage in this part of the cell. So how we do this? Molecular pixelation has 5 main steps. They are very easy to run. You can run them with commodity hardware that you have in your wet lab. The only part that it requires a specialized hardware is the last step, where once you have prepared the library, you want to actually sequencing it. There is no optics involved. There is no specialized equipment. Everything is done by DNA. So the antibodies have oligos, and all our reagents are DNA-based, which is a very elegant solution that we have innovated on. So everything is described in our recently published Nature Methods paper, so I encourage you to go and read it if you want the nitty-gritty details of how the method works. Here, my colleague Will and I are here to respond to your questions, so we can definitely give you a rundown on how the method works. And the method not only provides abundance of proteins, but it also provides the spatial distribution of the relative location of the panel of proteins that we are studying. And then also, we can study the colocalization of pairs of these proteins in a high throughput fashion. So we have packed this into a reagent kit for everyone's use. And we only require cells in suspension, human cells in suspension. So that's why we started studying immunology. We think immunology is really cool, and everything is immunology right now. We have a panel to start with of 80 proteins. And then you can run our kits. We have different 8 reactions, and it's 1,000 cells per reaction. And there is no specialized equipment required. And from cell prep to sequencing, you can do it in 3 days. And then to process our data, we have created our NF Core pipeline, or the NF Core pipeline called Pixelator, that I will present in the following slides. And obviously, this is free to use for everyone. So why to study the cell proteome? So cells have a dynamic distribution of proteins, and we can study different modes of distribution in a high throughput fashion. And this is really important because this is the way that usually cells respond to stimuli. So let's stop here and let's make a poll. So who thinks that the cell in the right is migrating? Raise hands. Yeah, right here. And who thinks is the cell in the left? Raise hands. Right. So we have a majority here, and you are right. So it's the cell in the left. We subjected cells to chemokines in order to start migration. And the important part here is that proteins are exert the execution function in the cell. But not only that, they also collaborate in different constellations in order to produce, for instance, cells to change shape and to do all sorts of different cool things that they do. So this is our latest application node, or latest piece of data, where we have subjected 3 different donors, actually, to activation by a common activator called PHA, and then compare them to resting cells. And we will publish these data sets also shortly. So on the left UMAP, you can see the 3 donors grouped into both resting and activated cells. They cluster the cells very well. But what is interesting to see is that if you look just at abundance, in this particular protein, that is one of the most polarity score ones, then you can see almost no difference. But if you look just at the polarity UMAP, then you can see that actually it clusters pretty well into the activated population. And if you take actually like just querying by cells, if you take like a resting one and activated one and you subject that to like a local scoring method, then you can see those clusters of this protein in the cell membrane being actually in microdomains that we can call out. And this is really cool because we took microscopy pictures, and then we could recapitulate this, actually. And if you look at the colocalization metrics, then you can see different constellations of proteins that they actually cluster together in pairs, either colocalizing or actually in blue, segregating as well. So they might displace each other because that's what the cell does. Like they occupy the space, so then other protein has to go out. And this actually has been also confirmed by super resolution microscopy. In this particular case, these 2 proteins, they actually colocalize in lipid wraps. And these are very mobile parts of the cell membrane in cells. So this is actually really interesting for us because we show this both in this case it's only polarization, but both in polarization and colocalization, we saw this orthogonality measurement between abundance and polarity and colocalization. So we think that they are measuring different events, and then there is information in all these different modes. And this was really interesting that our technology was working as expected. And also that it was very heterogeneous response when activating T cells with PHA in 3 different donors. And this slide is just to show different applications that our customers are thinking of. It's just things that either we have done in-house or someone else is trying to do, and also like the potential that the technology has. Right, so NF Core Pixelator. We are very thankful to NF Core and Nextflow. It has been a cornerstone part of our technology in order to ship to our customers in record time. When we started the company, it was basically a Python package and lots of different command line tools. And the reason that we have kept using Python for the logic is because we are trying also to maximize the amount of developers that you can put in the problem, on the intelligence of how to create new algorithms for MPX. We think also that at some point we decided to package these command line tools into Docker. That was a good way, actually internally, to commoditize that and to ship to different platforms. And then we actually glued that with Bash. But it was actually NF Core that, with the nice features that it has, that provided us to scale this process and actually ship to lots of customers and lots of different sites. Right, so historically then our pipeline that was presented in Barcelona last year is very simple actually compared to what you normally see here with lots of different tracks and really complex subway maps. So subway map is just going from filtering and processing reads to constructing the cell graphs and then annotating them and qualifying them into cells and then creating the different spatial metrics, calculating them and putting everything in what we call our pixel files, which is our data format that is also open. So we want to keep this complexity low. Actually, we don't think that it should be more complicated than that. And but we love certain features of NF Core. So actually, to start with, we love that it's super easy, extremely easy to deploy. It is really nice that there is all these different institutional profiles that work with most of the institutes out there that we work with. We love the plugin system as well, and also we have been lobbying that it also has these capabilities of running offline that is really easy to use and to explain to our customers. But we love working with the community, with the NF Core community as well. It has been extremely helpful interactions with Phil and the core members there in order when we decided to press the button open and say, okay, this is going out, this is coming out to the community. And it was like an extremely easy process that I definitely recommend. And it's something that we keep to our pride, like being the first biotech that are doing this in the open. And I recommend it to others. The only thing, maybe my recommendation is to think how you want to scale this process in the future. So actually, we love keeping features open and developing in the open for current products, and then having a process that we can develop future products in a stealth mode that then we can bring outside to the community. So this is our software site, and here we produce Pixelator, NF Core Pixelator. We document our software, but then also we explain with data tutorials how to analyze our data. And then we have a series of data sets that we actually ship in a Creative Commons license form. So you can explore the data sets, you can use the tutorials on them. Our Pixelator is also released under an open source license. The tutorials is kind of like a step-by-step journey about how we started, and we currently do still our data analysis process. And we hope that our customers can expand on that. And we will definitely grow there, and I think our support team would love to hear about your feedback on them and how what you would be interested in doing. So please visit our websites. There is a lot of information there, and our support team is excellent, and I think that they would answer any question that you might have, and I think tomorrow Will and I will stay here as well. So looking back, we started the company in the end of 2020. We started working with MPX in the beginning of 2021, and we started shipping kits on September 2023, if I'm not mistaken. And we could have never done this that quick without Nextflow and NF Core. We're a bit north now of 25 employees. And people say it takes a village, and it really takes, and I'm really thankful to these people about creating both incredible products in the web and the dry lab as well, so thanks to all of them. Thank you.
Thanks, Alvaro. It was a very interesting talk. We have a few questions here. So the first one is, what are the advantages of molecular pixelation over more traditional methods like FACS?
Right, so that question probably it goes into the application node. Like with FACS, usually you detect abundance in proteins, the intensity that you measure. Obviously, you have like a really high throughput that we cannot achieve at the moment, but I think that with FACS, you cannot really get the polarity or the clustering of the cells and the colocalization as well in a very high throughput fashion. Like obviously you can query in FACS a few pairs of proteins.
How have customers responded to you providing an open source analysis pipeline? Has there been any feedback?
Yeah, it has been really good. We actually have kept that private at the moment, so we have certain testimonials in our website, but usually the people appreciate it a lot. Like sometimes you go into conversations with customers and then you say, hey, you have a Nextflow pipeline, this is great. And then everything, it gets very, very nice conversation from now on. And also like the tutorials, they have appreciated a lot, and we recognize pretty early on that we have two communities of analysts. Like most of the people either talk Python or R, and then we provide the packages for data analysis in both.
Okay, thank you very much.
Thanks.